but no, hey, it's really great to meet you guys, and kudos to you guys for really doing this sort of translational career development program, because I think it's, you know, as you see, it's such a great way to do highly impactful science and medicine. So what I'm going to talk about is mechanisms underlying immunotherapy efficacy in renal cell carcinoma. And this talk is a little bit different, right? So, you know, I was kind of asked to talk a little bit about our own work, but I'm going to talk about sort of some of the general thought processes going on in the field. You know, what's approved, some of the things approved for other you know, indications, pan-cancer approvals, and then I'm going to do a deep dive into renal cell carcinoma. So a lot of this work is ongoing. Some of it's published. All of it is in partnership with, you know, a very close-knit team that we used to work with. Ari Hakimi used to be in my lab as a semi-independent investigator, and then Bob Motzer, and now we're working with Moshe, and of course Brian collaborating on some things, and at the Cleveland Clinic. So some of this will touch on that. But most importantly, I'd like to sort of focus on how mechanisms can inform us on how to design next generation trials. Okay, and as you know, based on a lot of work, including, you know, some groundbreaking work from Brian here on some of his trials with Axi and so forth that have been run, you know, I think you guys all agree that renal cell carcinoma right now treatment is undergoing just amazing revolution. So, and I'll summarize that and talk about what we can learn from the trials. So these are my disclosures. So this is no surprise to everybody here. You know, it wasn't so long ago that we used to think that tumors were autonomous beasts here, you know, growing by themselves, and we would focus only on the kinases or the tumor suppressors or what have you inside the tumor cell. But then, you know, it took a while, probably around 15 years ago, but concomitantly over many publications over time, including descriptions of some agents that actually now work in the clinic, that people started really paying attention. Just as sort of a bit of history, you know, we were part of the very first TCGA projects, okay? And part of that was delivering, you know, putting together tumors. So we're part of the tissue procurement stuff. And I remember distinctly the manual would say, well, you know, we want to throw away any tumors didn't have at least 70% tumor nuclei. Right, so that for one thing is what the TCGA samples are. Okay, so you know, if you use them in your analysis, just be aware that that's the case. But in hindsight, I'm just speaking for myself, feel awfully dumb for doing that, right? Because, like, you know, maybe it's just because we just didn't know. But now we realize that, as you see here, the tumor ecosystem is very complex. It involves not just tumor cells, but the ability of the tumor cells to grow is really influenced by tumor immunosurveillance, immunoevasion by things like T cells, Tregs, and K cells. Antigen presentation is really important. So it's really this whole ecosystem that's important. And the two top drugs, without getting into the nitty gritty, that actually have made an impact in the clinic are PD-1 and CTLA-4. You know, these are the main approved and things that target similar things. LAG-3, of course, anti-LAG-3 was recently given the go-ahead in certain circumstances, but for the most part, this is what you have. In the case of CTLA-4, as you know, we are inhibiting the inhibitors of signal 2. This is going to come back over and over again. Remember, signal 1, if you'll remember, is the MHC presented to the TCR, and this is responsible for either amplification or effecting killer mechanisms. And CTLA-4 B7 and CD28 system here really controls signal 2. Anti-CTLA-4 relieves the inhibition here, activating undergic T cells, right? So nivolumab's similar, but thought to be at the site of whatever you're looking at. It's very interesting. PD-1 actually, it turns out to be something that's critical for peripheral tolerance. Okay, so peripheral tolerance in humans is actually a really big thing. We were taught in immunology, of course, that central tolerance is the end all be all, right? So during thymic deletion, double positive, you delete everything, end of story. Not true. About 30% of all clones can be autoreactive, and they are silenced or exhausted using this mechanism or other mechanisms for peripheral tolerance. And that therein lies what the tumor cells take over and use. Very, very interesting, because it also ties into what types of secondary side effects can result when this happens. So these are the two main things. Together, these, again, Brian knows this, we cannot underestimate how much this has revolutionized treatment, right? When we started oncology, renal cell carcinoma mets, yeah, good God, you just can't get rid of them, right? They're just like rocks. And now you see these wonderful trials showing an amazing level of response. But the history-wise, it starts back with melanoma. This was one of the—
Can I ask a question about that previous picture?
Sure, yeah.
The one on the left, is the dendritic cell the only cell that will engage with CTLA4? Or is this—
No, this is a vast oversimplification of what is going on. But the CTLA4, the dendritic cells, APCs, also, this is also on Tregs as well. So you get a positive and negative signaling system going on. It's complex. But more complex is PD1 and PD-L1. That's on everything from tumor cells to regular cells to macrophages to NK cells and everything. So this is a vast oversimplification of what's going on. We think that this is happening, but in fact, this model here, you can see my cursor, right? This model here right now probably is not nearly what goes on for anti-PD1, because it's well known now that in tumors that respond, the clones that are originally present in the tumor, the TILs, aren't necessarily the ones that actually elicit tumor killing. In fact, there's some nice data from Howard Chang, some data from us, many others showing that the T cell clones, the effective ones, are all recruited from the outside. So just because you have a hot tumor does not mean, again, that you have effector T cells in there that are responsible. And so what that means is you have these guys everywhere, right? Outside, not here in the tumor. And that fits in with what we're thinking about peripheral tolerance, of course, right? It's a wide scale mechanism. So, okay, so you guys may know Checkmate 066, a little bit more of a historical trial at this point, but this was just mind-blowing at the time when it came out. This was nivolumab, anti-PD1 from BMS, that was, you treat, melanoma, metastatic melanoma with anti-PD1 versus dacarbazine, which is standard of care at the time. You see these curves were just not even close. So as a single agent, what has been really exciting about immune checkpoint blockade is that at least the anti-PD1 side of things has been active across cancers. This is a patient from Paul Chapman, a former colleague of mine at MSK. And this was really striking because you see here that this poor woman had a metastatic melanoma. She had a big tumor in the chest wall here. And after a single dose of ipi-nivo, the whole thing disappeared. All the tumor in her body disappeared. And while this doesn't happen in nearly everybody or as many people as we want it to, it is quite amazing. You never ever see this, really, right? And it was just super striking to us at the time, which is why I think it was submitted as a case report. But if you actually look at overall response rates as a single agent across multiple cancers, you see that you get some pretty good response. Pretty rare for something to sort of like push the envelope like this. Renal cell carcinoma is here, pretty decent response rate as well. You have a few things here down here, like Hodgkin's disease, that are really outliers. Here, it turns out that on chromosome 9, where PD1 is, what basically happens is that Hodgkin's disease is actually addicted to this. Okay, so it turns out that this is actually a lesion that in Hodgkin's disease is utilized for Hodgkin's disease to promote tumor growth itself. And that was a great proof of principle, but it also means that Hodgkin's disease, being particularly sensitive to anti-PD1, means that folks that are chemo resistant now really have an option. And so this has made a huge dent in terms of how many people with chemo resistant Hodgkin's disease who have avoided stem cell transplant. It's just really quite amazing. So the death rates from Hodgkin's disease now just plummeted. This thing's, essentially nearly everybody's cured, which is great. It's wonderful. So who responds and who doesn't respond? I've summarized this here for you. These are some of the major FDA approvals. The reason why I mention this is because we're in a time when understanding what happens in clinical trials is one in the same as understanding how to design the next generation trials. And you know, for the fellows and scientists that have grown up in this era, it's pretty amazing. Previously, as Brian may attest to, these were two separate worlds. I cannot tell you how painful it was to convince folks, like say in the year 1999, year 2001, and so forth, to get big pharma to actually pay attention to correlates. Remember, Jeff Engelman and others took almost a decade to get enough samples from EGFR lung cancer treated patients to study enough samples to study acquired resistance. Okay, so you know, and now you see that almost every single large phase 2, phase 3 study, definitely phase 1 studies are being sequenced, which is amazing. It's a great opportunity for translational scientists. So here, the fruit of that is this. Relatively quickly, we were able to understand some of the main factors that influence immune checkpoint blockade. And you have mutation burden here, which is approved. You have PD-L1, of course, which is kind of weird, but some of it is approved, and we can talk about that. You have things that generate more tumors. You have things, tumor rejection, stuff that generates less tumor rejection and more resistance. MSI, of course, is approved. There are certain things like the microenvironment and innate immune system and microbiome here that really tips the balance in both ways. A lot we need to understand. The important point here is that looking at biomarkers and approving biomarkers, you know, just because you have a biomarker does not mean that it may be useful in clinic. You know, it has to be cost efficient. It has to be, it has to give an answer. Not, and predictive value doesn't have to be perfect, but it has to help the healthcare provider enrich, right? That's all that matters. And it has to be feasible in the clinic. So how we think about stuff, and this will be really relevant for RCC in a little bit, is really for tumor killing ability for immune checkpoint blockade to work, signal 1 is critical, as I said, right? And a lot of things affect signal 1, including cellular phenotype and molecular phenotype. And this has been this has been what we have always believed. So in the tumor cell, you have a lot of things here that can affect what's presented and how the immune system recognizes the tumor cell, which underlies the molecular phenotype. You have T cells that can undergo a lot of signaling changes and so forth. But ultimately, we really need to think about the genetics and the biology that's guiding activation of signal 1. That's critical. That's what we have focused looking at. And there's 3 components to the core of signal 1, right? The MHC, the antigens, and the TCR. And so essentially, a lot of what we are measuring in the clinic measures all of this. TMB measures this. MSI measures something here and what is presented here. PD-L1 measures some combination of what's active here. So you can think of this as sort of a footprint of how one can think about therapies mechanistically. And so early on, and this is not so relevant for renal cell carcinoma, but TMB, you know, we had part of this is we had sequenced Keynote 001 and found that TMB was important. I'm not going to go through the history of this, but this is some of the lung cancer studies that we had published from the lab in lung and also in melanoma. If you have a high number of mutations, you do better. And this was shown by a whole bunch of phase 3 trials. It's been relatively difficult to take this into the clinic, largely because of the confounders, as I'll show you a little later on. However, this was nice because Checkmate 227 got for first-line lung cancer patients treated with ipi-nivo. They were selected for high TMB, and the response rates were very, very nice. What ultimately brought it to the clinic was Keynote 158, which is a big phase 2 that accrued TMB high patients, and what they were able to find is that the response rates are higher. So there's been a lot of studies studying this, but what I really believe is that when you're only looking at one biomarker like this and not together with PD-1 or some of the other key markers or what's going on in RCC, you may not necessarily get the whole story. All right. But for what it's worth, this is actually pretty interesting because we asked the question of what, you know, the genetics, epitope diversity, is that important in all tumors? And the answer is a lot, but not all. So this is a large study we published in Nature Genetics a few years ago, looking at almost 1,600 patients. For patients that were treated with ICI, you actually see that you can separate by TMB patients that have done better. This is measured by MSK impact with a nominal cutoff of 17 mutations per megabase overall here. And then you have like patients that have never been treated with ICI, and we sequenced about 9,000 of them, and you really don't have any difference. And when you broke it down, actually, what we show is that for the most part, TMB actually really does matter for predicting response and survival. So if you go, this is a forest plot format. If you go this way, the association between high TMB and survival is good. What's missing from here is a couple things. RCC did not work for RCC, and I'll show you some more data from McDermott and other papers that corroborated that in glioma as well. It's interesting because this really caught our eye, right? This means that something else is going on with renal, and glioma was easy to understand because basically you get no responses in glioma anyway, right? So no biomarker, and this is what I tell people, no biomarker is going to fix your crappy drug for a disease, right? So biomarkers do not make up for bad drugs for certain diseases. So in glioma, 0 responses, of course, no change. MSI, which is rare in renal, but I've actually seen one at MSK. These patients do respond. Actually, Brian, I'm curious, have you seen with all the patients you've seen, have you seen an MSI positive RCC patient?
No, I mean, we don't test it that much, but not that I recall.
Okay, I'm just curious because it's people say it's rare, but I'm just wondering real world data levels. But yeah, no, I think we've seen 1 at MSK and 0 here so far. But again, if you're lucky enough to have an MSI phenotype in any type of cancer, it is approved. This is the phase 1 results from Luis Diaz when he ran his trial. He obviously took MSI positive patients in a phase 1 study, showed that tumor marker load correlated with MSI positivity, which is the blue guys here, and then this is the waterfall plot, and this is a survival. So we're kind of getting a feeling for how this all fits together, right? It looks like genetic instability is important, right? So all the responders, or a lot of responders, are here, okay? And then you have right, for different types of cancers. And you have the pediatric cancers and the low TMB thyroid cancers here, where there's not very much response. Coincidentally, this is where a lot of targeted therapies have worked. And so we need to sort of start thinking about sort of these big, broad strokes of cancer biology, what's going on, and perhaps immunotherapy, as expected, is targeting sort of this therapeutic index that is based off of non-self, right? It's the orange dot there, the epitope diversity, which is what it's targeting, and that's what the immune system should be good for, right? Something clearly is different with renal cell carcinoma. We've in the lab looked at every single possible variation of genetic alterations, and only one has panned out. We've looked at all the different ways you can define TMB, ERVs, indel load, all of that stuff. And things may be marginally predictive, but certainly nothing like in melanoma or lung cancer. But regardless, if there is a renal cell carcinoma patient and you do find these features, these are pan-cancer approvals, and technically you can give them. That being said, this is already sort of first line. Some of these treatments are already first line for renal cell carcinoma. So it's really, you have a few overlapping approvals here, but it's important to go through the tumor agnostic ones, right? So you have MSI, of course, and that's approved for Pembro, for Nivo, and for Ipi-Nivo. You have TMB high approved for second, third, fourth line patients that have progressed through previous therapies. And then you have MSI positive in the first line for pembrolizumab for colon cancer, that's MSI positive. This is now first line, okay, very important. This has replaced chemo. Now, to RCC, there's been a huge number of studies now. Where are we in RCC? So in RCC, initially there was the study looking at Nivo compared to sunitinib, which is anti-VEGF tyrosine kinase inhibitor. But now there's a whole bunch of combinations, and we're in the era where this strategy has revolutionized how we treat renal cell carcinoma. So here you have ipi-nivo versus sunitinib. You're bumping up the progression-free survival and survival. You have Axi+Avelumab, which you have, look at the response rates here, really, really great. You have Axi plus Pembro, which Brian Rini here ran, great trial, and you bump this up. Then more recently, you have lenvatinib plus Pembro compared to, and this was a 3-arm trial that was published with lenvatinib plus Pembro versus lenvatinib plus the mTOR inhibitor everolimus, and versus sunitinib alone. And you see lenvatinib plus Pembro came out on top again. And most recently, really nice paper here, you have Cabo plus Nivo, which again moved the needle. So look at this. I mean, it's a lot of great positive trials. Really wonderful for somebody like me to see this happen for something that was so deadly before. Okay.
Can I ask both you and Brian a question? And this is also in part because when I'm writing the significance section of my grant, I need to know why I'm still studying kidney cancer. In terms of durability, you're seeing all of these plateaus at the tails of these curves. For some of the initial patients, how many years out have they been, and how are they, like, is the durability great, or is this just a temporary response and then resistance emerges?
I mean, so ipi-nivo has minimum 5-year survival follow-up that was just published. And yeah, if you're in the responders, that's the initial, I think Bob may have been also the first author in the other one, but if you're in the responder, those responses tend to be durable. And there's a tail of the curve that's probably somewhere in the 20 to 30% range for progression-free survival. So probably the cure rate, I'm guessing, is somewhere around there, rough numbers. But that still means 70 to 80% of patients aren't cured and need more therapy, if you want to look at it that way. We're far from solving this problem.
Right,
the reason, because I mean, so I just wrote an NCCN grant, and I mean, it got funded, but one of the reviewers came back and told me, hard to imagine that what you're doing is going to match up to like immunotherapy or something like that. And they were like, essentially the implication was, why are you even studying anything other than immunotherapy in kidney cancer?
So my answer to that, and maybe Brian will want to add on, is one of the key things that broke this whole thing open was the addition of multi-agent, multi-target anti-VEGF TKI. Okay, and you know what's sad is that this was not the first thing that people have tried, right? So biology from decades showed that VEGF was a potent inhibitor of T cells, and to me, this was a checkpoint. This is an immune checkpoint, right? And so there's a lot of science to do. Second thing is here, you look at right here, just like what Brian was saying. I mean, you're plateauing out pretty low here, okay? And actually, there is a very nice graph in this paper, if you want to go look about, and also most of the other ones about durability of treatment, and meaning that of the people who respond, what does a Kaplan-Meier look like? And it does dive down to below 40%.
Okay.
So acquired resistance is really key. That's why I keep on harping on you and Moshe and Shilpa to collect every single acquired resistance sample you can get, right? Because it's really important. Okay, so again, so TMB does not drive this, all right? So if you look at the Modermic data, which if you haven't already, please take a look, great paper, and some of our own data, you see that that TMB with response and non-response does not segregate for renal cell carcinoma. And this is the David Braun study in Nature Medicine with the BMS-Nivo trials. This is sort of like a mishmash of different types of trials, like 009, a couple other ones that showed the same thing. And this is actually our MSK cohort. And I left that off for better effect here. For renal cell carcinoma, you see that overall pan-cancer, you see that there is a very strong signal, but with renal, it's just not there. So what is involved? Okay, so if you go back to and remember our initial picture of the green box with the T cells and tumor cells and the molecular basis of signal 1, we started looking at different things. All right. So one of the things we looked at is we asked was, well, does genetics play a role at all? And if TMB doesn't play a role, then we ask the question, okay, does the receptor for the epitope play a role? Does the HLA molecule does? And my current thinking is that it does, but the signal is not so strong, but you can see it. And so the HLA diversity, of course, is fundamentally a key because it is associated with pathogen resistance, the ability to fight pathogens, and it's involved in diversity of the immune system over evolution. And this is first proposed to be important through a concept called HLA heterozygosity advantage, defense by diversity. The concept here that, if you're more heterozygous, if you have many different types of alleles, you can present many different types of antigens and you can fight off more diseases. And so we asked the question whether this is the case in cancers, and through several papers, we actually show that there is a signal here. So again, what we're studying here with TMB and other epitope diversity studies is this, but here, of course, the receptors that actually are needed to present the antigens are also important here. So these are studies mainly done in melanoma, but what we show here is that at least in high TMB tumors, HLA diversity is important. This is when you have maximal diversity to class 1, and this is if you have less diversity. And if you put TMB in, it expands that difference. So these folks have high TMB and high diversity of HLA. And we go on actually to show mainly in melanoma that that these signals can be seen in multiple cohorts, including folks that are treated with anti-PD-1 in melanoma that are all patients and also fully heterozygous patients. And you can see that high HED patients, the top quartile here actually does much better. This is TMB, high TMB, and together they do better. And why is this? Because when you have high HLA diversity or high, a metric we developed called HLA evolutionary diversity, you can bind more peptides. And this is data showing that the higher your HED is, the more viral peptides you bind, the more self-peptides, and to some extent, the more neopeptides that you bind. So again, so here we're measuring TMB, here we're measuring HLA diversity. We tend to think that both are important. And we tested this in a phase 1b that was done in collaboration with Joe Lee, Bob Motzer, and Ari's fellow. And this was a Leva-Pembro trial that we worked on with Isai. And the results here were pretty compatible with the phase 3, but in fact, probably even a little better. This is the waterfall plot, really, really quite an amazing waterfall plot, really nice response rates. And if you look at PFS, if you have high HED, you actually do better than if you have low HED, and this is mirrored by duration. And what we're able to do actually in this setting was actually plot 2-year progression-free survival as a function of each patient's HED, and you can actually see a dose response that has occurred, which is what you would expect for a continuous variable. So if you look at the multivariate analysis, you can find that TMB does not have an effect in this trial. You have varying amounts of effect for a variety of other risk factors. The numbers are relatively small here, but you see that HED has a pretty dramatic effect on hazard ratio, improving survival. So maybe HLA has something to do with it. We then went on to look at the microenvironment. And in my mind, actually, renal cell carcinoma, for some reason or other, maybe because in certain organs you have immunosuppressive environments that are just critical to how the organ functions, right? We know the brain is extremely immunosuppressive environment, right? It has these microglia that just suppress the hell out of everything because you don't want T cells basically in the brain. That's called encephalitis, right? And so maybe renal cell carcinoma is something like that. We know that in pancreatic cancer, pancreas, pancreatitis is a huge problem, right? Due to all sorts of reasons, drugs, stones, alcohol, right? And the immune system and the tumor microenvironment is very, very suppressive. So maybe something like this is going on. I actually think, and so do a lot of other people, that this is probably the dominant thing in renal cell carcinoma that's going on now. You look at some of the other disease types like melanoma and lung cancer, and microenvironment is important, but the genetic potential of rejection is very strong, right? That shines through. Here, I think microenvironment is critical. And so we focused a lot of our effort in looking at the microenvironment. This is one great trial. This is obviously many people were involved, but Dave McDermott was the organizing author. And on his trial of atezo plus Bev, this is anti-PD-L1. This, of course, is antibody against VEGF. He began to see actually several transcriptomic subgroups, right? And these could be separated into different tumors having signatures. And I believe this was the sort of one of the first indications from trials that we're looking at sort of very different molecular subgroups that may actually impact treatment response. So when you actually look at these subgroups, you see that the sunitinib group is here. If you look at actually, groups that are positive for the immune signature, you actually see that the atezo plus Bev treated samples, actually they did better here, okay? And so they were able to identify preliminarily some signatures that may be involved, like this, what they all call the T effector signature. One of the things that we started doing in the lab is this large project in collaboration with Ari that was published. And I think that this delves into how we were trying to solve this question. And it's fun to actually go through the logic of this, right? So this was a neoadjuvant trial. And so what we all did was we treated a number of patients neoadjuvantly. And some patients responded well and some patients did not respond well. So what we did was multi-regional, multi-omics. We actually mapped out the kidney, and we basically worked with a pathologist to set up a 3D map of all this. And within each part, we took cells for a bunch of genomics, including single cell genomics. So it's basically multi-regional single cell. And the reasoning is behind this is because if you actually look at gross sections of a responding tumor, you actually see a lot of heterogeneity in terms of response. Some areas respond really well and some don't. And you see that actually this is a big issue for kidney cancers, at least under the microscope. And so we want to understand, okay, within one patient, what exactly is going on? So if you guys haven't done single cell allows you to actually analyze the transcriptomes of individual cells. So we had this was again circa like 2018, 2017, before single cell really started getting going. So we had to work out a lot of different things, but thankfully now it's better. And we were able to actually throw in TCR sequencing at the same time, which allows you to analyze sort of the diversity of T cell receptors going into different places. So this is a lot of fun. The bottom line behind all this is that you see some very, very interesting patterns. You see from single cell analysis, you can find your tumor cells, your normal cells, of course, you find your blood cells, and you find your lymph nodes, and you can quantitate where things are. But you also see these, saw these really, really interesting groups that didn't occur in normal cells. And here, specifically, we have this CD8 tissue resident T cells. Now, around the same time, if you'll remember, there was a lot of work going on with this concept of sort of organ-based, site-specific T cells. These innate, they're called innate lymphoid progenitors, some people call them, right? Or NK-like T cells. A lot of people have given them different names. And in fact, that's kind of what they look like. They're kind of in between NK cells and in between T cells that are tissue resident. Okay, it's very interesting. And the bladder group from UCSF had shortly after saw the same thing in bladder cancer. Really interesting. So it's this purple group that we saw that was really unique. And of course, we saw the main tumor types. It was very important to be able to separate tumors from normals. And one of the important things to remember when you're doing type of this stuff is you want really good controls, okay, as you're designing your own experiment. You want to basically always make your libraries of all the different parts of one patient all at the same time. Otherwise, you'll never be able to deconvolute some of the batch effects, okay? So that's critical for examination of this. And then we went ahead and looked at all the different types of cells here. And this is a little bit complicated, but you see all the different types of cells here as a function of response and non-response. And again, you see this CD8 tissue resident signature pop up in our complete responders that were much higher in other places. You see the mixed response folks did have the exhausted T cell phenotype, right? So they had a mixed response, but also a little bit of this, but you see that the complete responders, this really stuck out. And what they look like, we did some trajectory analysis to figure out exactly what this stuff is. And it seems to be a trajectory of differentiation, right? So this, we blew it up here and separated out as a function of response and resistance. And it turns out that if you have a differentiation of these guys in response to ipi/nivo into an NK-like phenotype, so your trajectory goes this way, green, you respond very well. And that's in line with a lot of what other people have seen now too. That it's not necessarily the phenotypes of any old standard CD8s, but this is likely something that exists, that sits there already, or gets recruited, but has this phenotype of NK-like. And if you're resistant, or if you have a mixed response, what happens then, these same cells can actually go down and become exhausted. Not all of these guys here go down this route, this green route. It can go this way as well. So I suspect that looking at these types of mechanisms are going to be key, right? Because where are your next targets going to be, right? You're not going to just randomly pick stuff off, like the Chang and Melman, list of different immune synapse, markers. You're going to be looking at markers and targets that are critical for what is necessary for response. So what does the T cell resonant signature look like with Axi+Avelumab? We took that signature and we also applied it to a variety of other data sets, including Emotion 151, which again is the McDermott, and actually you see that this is our signature here. It's really performs better than any of the other signatures. And in Javelin 101, Axi+Avelumab, we see that worked here as well, whereas it didn't work in the sunitinib arm. So really kind of cool stuff. What does the signatures actually look like? It's an amalgamation actually of a lot of other signatures, right? There's this signature that includes memory phenotype signatures as well as CD8 activity. It does have somewhat of a effect on the TCGA stuff, but actually, this is what the data looks like for compars, which is pazopanib, and sunitinib, and so forth. So, it kind of fits in with, not so much with when immunotherapy is not given. But when immunotherapy is given, which is like with the McDermott study, you see that you can actually see this change. So this is the last couple of vignettes I'm going to show. This is a key study that I think everybody needs to know, and I think Brian, you're on this paper as well as Bob and many others. This was a study of a whole bunch of different renal cell carcinomas on study. And they were able to identify several clusters through a non-supervised method. And these, again, really are important because they provide implications on what the next targets are. I think this very well sums up what a lot of folks, including our own data, McDermott, the Dana-Farber data, all showed in a more piecemeal fashion. This showed in an integrated fashion that there are these major microenvironmental drivers, these TME subclasses that actually are critical for trying to choose what types of therapies. And essentially we're targeting the microenvironment here, right? Which is kind of a different paradigm than what we're used to, which is targeting the cancer cell. So I think these subsets are going to be critical. You have an angiogenic subclass, which is very similar to what we had seen before. We had an inflammatory subclass. and so forth. So we have an angiogenic one here. So moving forward with all of that, what does identifying the molecular drivers of response, how is it going to look like in the next 3 to 5 years? Okay, I can assure you that it's likely going to be multiple factors. We're heading into, I think, a time when we're going to be start using for immunotherapy a model that's similar to breast cancer. And those folks have been using nomograms for a long time now, right? With adjuvant online, prostate cancer with the Catan nomograms and so forth. And we're not going to be able to just use one factor because anti-tumor immunity is something that is driven by a complex biology, and naturally you're going to find several markers. And so we underwent a study recently just to figure out what those are. And we want to address a couple things. One, we wanted to train whatever model we developed in an unbiased manner. We put in a lot of different factors, both genomic as well as clinical, everything from labs to age to sex to mutation load, what have you, right? And then we use machine learning to generate a model, and that model was trained actually off of response. It was trained off of radiographic response. We actually got a consistent reads for over 1,000 patients. It took like 2 years actually, and we trained against response because survival is often confounded by a lot of things, right? If a patient gets resistant, comes off, your survival may be different. It's really hard to control for all that kind of stuff. But response within 3 months is, either the tumor shrinks or it doesn't. And what was interesting is that we found a bunch of things that we knew and a bunch of things that we did not know. So overall, we developed a 16-factor model, and this includes RCCs in here, by the way. It works for RCCs very well. And the prediction was actually pretty good. It was like 0.85 at this point. Training was 0.83, all above, almost close to 90%. And one of the things you can do in this random forest model is actually look at feature contribution. What factors are most important? And the top 16 were located here. It turns out the TMB is very important, as we knew. But it turns out that chemotherapy prior to immunotherapy was the second most powerful factor. And it was not chemotherapy during immunotherapy or chemotherapy after immunotherapy. It was specifically chemotherapy before immunotherapy, which is really interesting, right? That's how the DERVA trial, Pacific trial, was run, and that obviously changed the practice for lung cancer. But also you find things like albumin, pretreatment albumin, age, platelets, just basic labs that are really interesting. Albumin was particularly interesting because we know that albumin is associated with general wellness, nutrition status, and so forth. But to find that this is the number 3 factor associated with tumor shrinkage was very interesting for me. And HED does have a signal, but it's within a larger context of other signals. By the way, MSI here is not very positive just because there were very few MSI patients in this cohort. So this takes into consideration numbers as well. And so we generated a comprehensive model here. It works pan-cancer, works for melanoma, works for lung cancer, works for others. This is a one-size-fits-all model based on the combined training set. I think that if you retrain those factors for each individual tumor type, you can actually get better optimization. We have unpublished data training this for renal cell carcinoma, and you get a nice separation. It's not published yet. We'd like to get a separate validation set, but that would be something really, really fun to do, perhaps in collaboration with this group. One of the things that's interesting is if you look at up here, BMI is important, right? So that's interesting because it's been hard to quantify the ad hoc observations that people have seen. People have noticed that obese patients do better, and we were able to validate that. Turns out that if you have all comers here, you're obese, you do better. You're overweight, which is in between, you do a little bit worse, and then normal, you do better. But the signal is dependent on what type of tumor you have. And renal cell carcinoma is the number 1 driver of this factor, actually. So this is response here of obese. And then this is if your BMI is less than 30. And there are a few other tumor types where there's a little bit of a change here and there. like mesothelioma here, but this is by far the biggest driver of the signal that we see. Very interesting, right? So is it a coincidence that, you know, the renal cell tumors are always surrounded by perinephric fat? Is there something that's related to all this? I don't know, but really, really intriguing here. And look how strong these effects can be, right? So if you have TMB, right, greater than 10 and less than 10, using the nominal cutoff from the FDA approval, you see a nice stratification. But then if you segregate secondarily by BMI less than or greater than 30, you are actually shifting the whole curve here. So what's important is, as we move forward, especially in clinical trial design, I really suggest that we, at least in terms of variables, we really start looking at more than one variable. It's really critical, right? So if you just look at anti-PD-1, look what a mess that has caused in the field, right? Just look at one PD-1, you're confounded by all of this, right? So so I think it's very important as we move forward in our trials. So again, very important paper. I come back to this largely because we see now targeting the TME here is really critical. The TME is shedding light on what potentially can be targeted here, whether you want to prioritize angiogenic compounds, whether you want to target prioritize checkpoint inhibitors, like with this immune active subgroup here. I think that this gives us sort of a roadmap specific for RCC. And in fact, a lot of that is already happening, right? You see all these agents that are in play right now. This came from a nice review that just came out in Nature Reviews Urology. You see things that actually target the immune system, but you see things that also target some of the things that are on some of these categories, right? You see things that are targeting the cell growth itself. You see things that are targeting the microenvironment. Some promising avenues that I think are critical that we look at include more advanced and better ways to target the myeloid compartment. If you look at these renal cell carcinomas and look at some of the signatures that I've been talking about, myeloid weighs heavy on this tumor. Myeloid polarized the wrong way can be very, very immunosuppressive, and I think that's what's going on here, right? So I think new agents above and beyond CSF1R inhibitors and interferon gamma, PIK3CA gamma inhibitors are going to be critical. Adenosine targeting, for instance, also can be important. So you're going to have these slides, so I'm going to skip this summary and leave time for questions, but a ton of stuff, at least for the part that I presented from our lab. Ari Hakimi has been really central to this group, driving a lot of work here. Bob, obviously, at Memorial Sloan Kettering, and collaborators here at MSK. Thanks so much.
Thanks, Tim. Really appreciate it. Great summary. I'll open it up to the group, for questions, comments, anything.
And related to your question, Abhishek, I mean, I think renal cell carcinoma is one of the best things to study right now, right? It's not, this is exactly what you want to see, right? You want to see a convergence of therapies that are hitting and shifting the curve on a lot of different grounds, right? You have a lot of different avenues. Like, look how many things that you could propose, right? Look at that. And all these things have solid preliminary evidence and preclinical evidence supporting some efficacy, right? So to me, it's super exciting. We've slowed down a little bit with RCC. I know that Brian is working with Marcella on some studies, but again, like, it's a great opportunity. I think we really got to prioritize some projects and just really push. And it's a lot of fun too. So
I have two questions actually, Tim. So one I think is a more concrete question. The other is a thought question. If we try again, and coming from my perspective of trying to model the disease in experimental systems where we can address functional relevance, how does the HLA diversity in the humans, what would be the right way to model that in mouse models? Are there like animals with like MHC diversity that can compare to HLA diversity in humans?
Yeah, if you want to, undoubtedly nothing gets rejected without a genetically based epitope, right? So buried there somewhere beside, once you get through the microenvironment and all of this, that's where your genetic rejection potential is actually going to really be manifested. And what those genetic rejection potentials are, I don't know. But what I do know is that MHC is important for presentation. That's what the MHC data shows us, right? Where to go?
No, I'm thinking more specifically, if we were to, for example, take the Brugerolus mutations or the Fru mouse model mutations, and could we now be crossing these into animal backgrounds where those strains have high MHC diversity versus some that don't, and now start actually in real time modeling the impact of MHC diversity in animals to therapeutic responses?
Yeah, there have been some papers after our initial paper on the MHC LOH, which is the older one, came out. Actually, David Baltimore, actually of all people, took his mouse lines that he had and made basically ones that lacked one allele. They were heterozygous. They had LOH on that area, and it recapitulated this phenotype. So you could make something similar with RCC, and then you can try to measure that phenotype. And you may want to ask the question of, okay, well, how hard do you have to hit the TME before you start seeing the effect of epitope diversity, right? That's a fair question.
Yeah, I think that would be fantastic, especially if we start tying it into some of the new studies that we are doing with, the microbiome's impact on the TME, and also biological sex-specific differences, etc., things of that nature. I think those models will become really valuable.
Yeah, go ahead.
I was going to just come to the last picture from the review that you had with the targets. I think historically we have tried to make our TKIs and try to scrub them clean of quote-unquote non-specific targets, if you will. Honestly, I don't know if any of our TKIs are really truly that specific. Probably like what we think is hitting VEGF is also hitting probably 10 other tyrosine kinases. But maybe we should start thinking in terms of more broad spectrum TKIs now, like things that we discarded because they were hitting more than one TKI at the same time, and then ask the question whether they would engage multiple relevant tyrosine kinase at the same time and then synergize better with the checkpoint inhibitors, right?
Yeah, I agree. This here is not entirely accurate. This shows some of the larger propensities to target, but I can tell you that lenvatinib hits all three of these.
Yeah, for sure. Yeah.
So so you're right. I think all of these are multi-agent kinase inhibitors, and I don't believe that it's a coincidence that these have worked much better than Bev, right? So Bev hits only VEGF, and that does not work nearly as well as these guys. So I actually think what we're probably doing is hitting a combination of these things, but we're also probably hitting macrophages with this. PDGFR receptor is very high on macrophages, right? And it's suppressive, right? It's immunosuppressive. So macrophages also, in addition to T cells, also have VEGF receptor, right? So I think you're right. I think that the relative dirtiness of these kinase inhibitors are really important.
Yeah, I mean, I think the dirtier ones are probably more active, but also more toxic. I mean, Lenva is a really tough drug to give, and Cabo even. So I think there's probably some sweet spot of, multi-targeted enough, but tolerable enough and dosed well, and, whether some of them are more that play better, some of them are more tolerable with immunotherapy, whether the dirtier ones are quote unquote better, more additive or synergistic. I'm not sure there's hard data, by the way, but I wouldn't doubt if that's the case.
One of the things I'm interested in is, you have these things that are targeting the microenvironment. I mean, this is primed for putting in sort of more tangential treatments like cancer vaccines, for instance, right? That have a different toxicity profile that can help you drive this. Because once you sort of soften up the microenvironment, you can do a lot of other different types of things.
I mean, I will say that I think, clinically, we'd like to move away from VEGF TKIs. I mean, they're pretty toxic and need to be chronically administered. And I think their biggest utility, frankly, is just in upfront disease control. And I think if we really want to help patients long term, we would use them up front and then drop them and then really build multi-agent immunotherapy regimens. And there's some movements of that, but unfortunately, like BMS is a good example. Like, you know, they're sort of the immune company, but instead of building on their own immune pipeline, they went and combined with cabozantinib, right? And that's, I think, going to turn out to be a mistake, et cetera. So, you know, we haven't really done a good job of that. But if I were king of the world with unlimited resources, that's what I would do.
So I have one question. Sorry.
Yeah, go ahead.
So yeah, so as you said, the data you showed, like the combination works better, like chemotherapy versus immunotherapy. So first you hit with chemotherapy, then if you put immunotherapy, it works better. So how to do those things in mouse? Because it really means it's patient, it's easier to do that. For mouse, like we always usually treat the combination at the same time, because like if we hit for, like, let's say chemotherapy for 2 to 3 weeks, and then we wanted to do other one, the mice dies in between. So that's one of the possibility problem in animal models. And also I'm thinking another question is that recently, I think most likely 2-3 years back, one paper came out in melanoma. They actually did some analysis of immunotherapy resistant patients, and they showed that some angiogenic signaling is higher in the immunotherapy resistance patients. So do you think, like, following up with an anti-angiogenic therapy, following after immunotherapy will be better instead of combining them together?
Well, yeah, I mean, it is true that a lot of acquired resistance, the phenotypes are not what you would expect. So, you know, when at the beginning of sort of the modern period of time, when people started really looking at this, you know, from a genetic point of view, you know, me and maybe some other people had thought that some of the obvious targets would be the source of resistance, like deletion of beta-2 microglobulin, right? But it turns out that that doesn't happen very much. And it turns out that from what we do know of acquired resistance samples, and like you said, mainly through melanoma, right, is that, yeah, I mean, the T cells can be very high in them. And it's not a question of T cell engagement or APM activation. It's a question of something else going on. So myeloid actually is one of those things. If you look at resistant tumors in RCC, they're dominated by even more myeloid cells. Okay, so I think myeloid is important. But in some cases where myeloid is not so important, like melanoma, I think it's got to be multiple checkpoints that are upregulated. One of the things I didn't talk about here is we did do a trial with BMS, this pure biomarker trial, in which we did biopsies before and during treatment, right? And what you can see reflexively as part of the effect of the drug, the pharmacokinetics of the drug, is a reflexive activation of immune checkpoints, many immune checkpoints. So you treat with PD-1, anti-PD-1, and in the in all patients, about a dozen immune checkpoint blockade molecules get upregulated. Okay, but in the ones that become resistant, what happens is some of them become really high. So there's something going on with feedback loop here, and if you get a reflexive upregulation due to feedback of other immune checkpoint blockades, but that may be setting the stage for true resistance as the resistant clones begin to really expand and amplify that resistance. How does that occur? I don't know. It's got to occur somehow epigenetically. A lot of people have been looking down this road to combine with polycomb inhibitors, EZH2 inhibitors, these kinds of things. But I don't know what the answer will be. A lot of these T cells have high tox. So eventually what happens is they become, checkpoints go up, and then tox and epigenetic program then completely locks down and shuts down the cells. So, and that happens in some patients and not others. Don't know why.
Hey, Tim, I need to run to start another meeting.
Okay.
Really appreciate your time. You're welcome here anytime. Appreciate all your input. Thanks everyone for the good questions.
Thanks so much. Take care.
Yeah, take care. See you soon. Bye-bye.
Thank you.
Bye.